Yeah. But some like yeah, it's just very different. Sometimes it's also feel very nice to be in those old building with the
Okay.
old charm. But I guess it's
Also,
better.
Yes.
Yeah.
So we'll do that.
Can you hear?
And then we can hide this. Yeah, so it's going now and it's recording as well.
Oh, it's now being recorded. Yeah.
Yeah.
Yeah, later we will trim the beginning.
Okay. Yeah.
And so
Okay.
the down arrow is going to be next slide.
Okay.
Yeah. And then that bottom if you feel like playing with a laser pointer.
Yeah. I'll see. Yeah. Hard to see, but anyway, yeah, I'll adjust it. Yeah.
And then this mic isn't on the laptop, but like
Okay.
it's speakers for the room.
The room is pretty small enough. I can.
Yeah.
I don't need to. Yeah.
Are the lights okay like this? 'Cause
Yeah.
we can
Fine, yeah.
also do.
Oh, so cool. Yeah. Okay, it's like a theater now. Yeah. Yeah.
Oh.
It's too complicated. Yeah. Wow. There's too much going on here.
Yeah,
Lights
this
pop
is a
up.
lot.
Yeah.
So there's all the projector and screens.
It's like yeah the cockpit now. Yeah.
But then it's pretty much done.
Yeah. Cool. Okay. All right. Thank you so much.
And then click on that.
Okay,
So
okay. Okay. Yeah.
perfect.
All right. Yeah.
All right.
Okay. Cool. Thank you. Yeah.
Thank you. Start around like 32 around here.
Okay, that's fine. Yeah.
Introduce you.
Don't worry. Yeah. Yeah. Just go. Yeah. So it's like an hour or 50 minutes.
Yes, yes,
Okay. Yeah.
yeah. And around 4:30.
About 4:30. Okay. Yeah, sure. Okay. Yeah.
Like I'm either like fully engaged or I'm fine. I do. Actually, just bought 2 more.
Wow.
Because it's Saints, and I can keep one at home, and I don't have to carry it with me. Yeah.
Hi, everyone. Welcome to the seminar today. So let me get started with introducing Dr. Hongyu Zhao, because I got quite a long introduction here.
Make it simple, yeah.
Dr. Hongyu is a professor in biostats and statistics and data science and genetics at Yale University. And he received his bachelor degree in probability and statistics from Peking University and PhD in statistics from UC Berkeley. And his research interests are the development and applications of novel statistical methods to address scientific questions in genetics, molecular biology, drug development, and precision medicine. And Dr. Zhao has served as co-editor of JASTA and Statistics in Biosciences, and he has received many distinguished awards, including the Mortimer Spielman Award for Top Statistician in Health Statistics under the age of 40, awarded by the American Public Health Association, and the Paulus Tsu Award from the ICSA. And without further ado, we're really happy that you're here, and we look forward to your talk.
Yes, thank you for a kind introduction and invitation, and thank you for coming and being great to catch up with like the White, who was here. I'm happy for him to be here. I'm also sad. Also, I'm meeting with faculty here and to talk about research and other things. So today I'm going to talk about correlations. You see, this is a very fundamental concept in statistics. And so probably some of you or many of you have seen this, the table, Francis Galton's account, how he came up with this idea of quantify the similarities between the mid-parents' height and children's height. And there's a very interesting article by Steve Stigler about Francis Galton's account of the invention of correlation. So like for a very simple concept that we know today, correlation, it wasn't simple for people to think about in the first place. It actually happened in his Galton's own words. It happened when he was actually commuting or just traveling from Oxford to Cambridge, and there's a rainstorm, and he happened to like a shed where he came up with idea. I mean, without a rainstorm, probably the idea could come maybe later. But it's a very interesting article to read. But it's not a fundamental thing. But you basically from the figure you can see the clear sort of the association between the mid-parents' height and the children's height. And so this is just like we always have different way to quantify it. So this is a figure to show the sort of the scatter plot. On the x-axis is the net income for a given family. The y-axis is the food expenditure. So you can look at the scatter plot. You can see, oh, there's definitely there's some positive correlation there, right? And the more people make, the more they spend on food. But also, as you can see, maybe hard to visualize as you go from left to right. When people make more and more money, the correlation, the dependence or the correlation between how much they spend on food, really, the degree of the sort of dependence becomes less and less. Like people have enough money, they have different ideas how to use the money. Right? So in that sense, you can certainly say you have global correlation to quantify, but also there's some local pattern there. You can say that the dependence may be stronger when people make less money, but the dependence may become stronger. So actually, this was my first paper I wrote about 30 years ago when I was a student at UC Berkeley. And so we came up with just local correlation there to really look at the quantified the local correlation pattern using a methodology that was very sort of new at that time, local correlation, local core correlation and kernel and to quantify and estimate that. So this says using a very global correlation may not be very comprehensive enough to capture the heterogeneity. In fact, this is so-called heteroscedasticity, right? In terms talking about the errors as a function of x, and so that comes to, in this particular case, the global correlation may not be a good way to measure or quantify. But today I'm going to talk about correlation in two specific contexts. One is the genetic correlation, and the other one is the gene correlation. And so at lunch, I'm talking to students to discussing the difference between genetics and genomics. And the first topic is more about genetics. The second topic is more about genomics. So it's very hard to tell the difference, but we'll see as I talk along. And in the case of genetic correlation, I'll talk about global as well as local, and to show the local correlation there can sometimes be revealing about some patterns that may be missed by the global correlation. And also in terms of gene correlation, I'll talk about the bulk samples or cell type specific gene correlation. So now just look on the first topic, genetic correlation. So genetic correlation is really, there's a term in genetics called a pleiotropy. So pleiotropy refers to the fact that one gene or one variant may impact more than one phenotype, right? That's for HLA, that has impact a lot of autoimmune diseases, and this is a well-known thing. And also the genetic correlation you can think of as a way to just think about these different families, the different like the net income, a way to quantify the global correlation. If you have many variants, each variant has effects on multiple traits, as a way to quantify the global correlation pattern to see how similar the effects are on different traits across all the variants, right? That's as simple as that. That's a genetic correlation. So we'll rigorously define what do I mean by genetic correlation. And since most of the gene people are not here, they're in Pittsburgh today. So I'll just spend a little bit of time so I don't feel too guilty to go over this basic concept now. Yeah. So the single nucleotide polymorphism represents the variations in the human genome, right? That's right, base pair difference across individuals, right? So this on this figure, there's a one base pair. There's two chromosomes. One chromosome carries C, the other chromosome carries G. So these are called the single nucleotide polymorphisms. There are actually there's 3 billion base pairs in the human genome. Actually, there's a billions of single nucleate where the single nucleate, not polymorphism, single nucleate variation or variants people have found from like different large sequencing projects. And these variants they have been found to be associated with a lot of different phenotypes through these so-called genome-wide association studies, right? The genome-wide association study is basically a very simple study designed to try to correlate or identify the SNPs or genetic variants associated with a trait. In this particular case, a binary trait. You can think about continuous trait, whatever you have the leftmost column is a vector of like the trait. And the other columns, there's a lot of like the millions of like variants or even more that represent the variations across individuals. Also, they have some other covariates. And the genome-wide associations come down nothing more than try to identify the correlations between the trait and some of the columns there that represent genetic variations. Right, so it's as simple as that. You have a lot of people collected, and right now maybe a few 100,000 or even millions of people collected. And you have a lot of phenotypes. You look at every single phenotype. You can look at every single marker, and then you do like a very simple association test. In this case, you can think about chi-square test or other tests. You get p-value. You make multiple comparison adjustment. You get a set of variants that are reproducibly replicated in different data sets. And then you call, okay, I have some ideas which variants are affecting which phenotype. So it's as simple as that. So that's GWAS. And the very first successful GWAS study was published by my colleague at Yale, Josephine Ho, back in like 2005, about 20 years ago. Actually, by really paying attention to the phenotype, and she was actually able to identify a major gene for the age-related macular degeneration. This is the very first GWAS paper ever published. It's one of the 10 science breakthrough papers a year in 2005. So it's only 96 cases, 50 controls. And she was only looking at, instead of millions of SNPs, only look at 110,000 SNPs. But it's sort of so-called the Manhattan plot that's looking at. So fast forward many years. So the kind of data that we are analyzing, the skills on discoveries, no longer just one major gene for a trait involving a few hundred people, but rather like hundreds or thousands of sort of the genome-wide association, significant results involving millions of people here. So in this particular case, people look at height. Remember the first So Francis Galton's idea, looking at the correlation there, the height is probably the most heritable trace. About 80% of height is due to genetics. And so, well, that's no wonder people like took on height as one focus to do initial GWAS. So this one involved about 5.4 million people in the study. The whole state of Connecticut has about like 3.5 million people. Right? So this sort of skill, and identified 12,000 independent SNPs in the genome. So this sort of the study gives a lot of ideas about what genes, what variants maybe are fighting the trait for height. Right? So it's a lot of GWAS results. And then on couple, if you have a GWAS result for multiple traits, you can ask the question like whether there are some shared genetics between different traits, right? We have thousands traits analyzed with many, many discoveries, and how to really quantify or identify there's some shared genetics and for different purposes, right? So it's a very simple idea on how to really quantify, how to create a metric that's really quantify the degree of genetic correlation. Is suppose we have the markers across the genome, you have maybe a few hundred, a few thousand, or a few million rows. For each row, we have the effect size for the trait 1 and trait 2, right? It's like, yeah, and then a very simple idea to quantify the genetic correlation between these two traits. You can just look at the correlation, right, between the effect sizes. That's it. Right? So this is as simple as if you knew the two effect sizes. So this would be very simple. Just do the correlation there. And in other GWAS studies, they report is the results using the summary statistics, where in this case, the first column represents the names for different markers, not SNP1, SNP2, but actually there are the reference sequence number there. And there's a little bit more information about the allele frequencies. And also B is the effect size, standard error, and the statistical significance. And the last column represents the number of samples used to make inference about effect size, right? And so this is all that we can gather from the GWAS, but the problem is that we don't really know the true effect size, right? So in the parentheses are the true effect size, but what we observe is only we can estimate from the samples available. So in general, there may be some discrepancy between what you observe, what you estimate, and that we all know this as a statistician, that's really how we make a living, because we don't know the truth, we have to make an inference about it. Furthermore, in genetics, there's another complication that these genetic markers, they're not independent, right? So think about like independent families, like this net income, so we can say the data points are independent. But in genetics, these markers, they are not independent, so they are positions along the genome. Some markers, for in particular case, I split this SNP1 into 4 different rows. In this case, on these 4 SNPs, they are in so-called perfect linkage disequilibrium. They are just tracking each other, right? 1, 2, 3, 4. So actually, instead of seeing 3 SNPs, you see 6 SNPs. And also, but you see there's a redundancy there. And this kind of dependence has to be taken into account to estimate. Even you know the true effect size, and you wouldn't count the first 4 rows as 4 independent rows, right? Because they are very dependent on each other, right? So this has to do with the design matrix, right? We know how these markers are dependent on each other, so we need to estimate that. So what to do about it, right? So we do not know the effect size. There's only limited thing we can do. So as a statistician, we have to resolve this. What can we do? So we have to rely on some models. And so I said, all the models are wrong, but some are useful. So the models actually that I'm going to talk about actually dates back to 1918 in paper published by R.A. Fisher. This sort of more like the thesis from his undergrad study from Cambridge. Was classical paper. It's a foundation for the whole field of quantitative genetics. And also, there's a few terms introduced in the paper we still use today. Variance, ANOVA, assortative mating. So it all came from that particular paper. It's very interesting, very intense reading for that paper. But the fundamental model in that paper is this one. It's additive model to describe the relationship between genetic variants and the phenotypes. And so in Fisher's term, the phenotype Y is due to the additive effects of infinite number of variants. That's just like before the M there, but it's a very large number of variants of fighting the trait. So it's a very simple additive model. Y, you have G1, G2. You think about a lot of variants in the genome, right? Each variant has some effect on the phenotype. And then there's the environmental factors, also some noise. So the effects from G1, you can think of like every single variant, there's a variation across the genome, right? Across individuals, right? For example, there's two alleles, there's three different genotypes, right? For example, there's A1, A1A2, and A2A2. There's three different genotypes. And we can call the genotypes as 0, 1, 2, right? And depending on which genotype of individual has, and then the effect size, you can say there's this U is the effect size. Now we normalize genotype score so that we recall the genotype so that the mean and the variance of each genotype is the mean is 0, the variance is 1, so that to make the sort of this more like equitable across different genotypes. Now the C1, C2, C3, and Cm, they represent the effect size on the phenotype. So this is a very simple model. But in this particular case, if I have a phenotype Y, we may have millions of things here. In the GWAS, actually, we are estimating the effect size of millions of SNPs. And if we try to really estimate every single individual effect, we know we're better off. So one thing that we can bring things together is to ask for help from the random effects model, where we assume the U, the effect size, the effect size U, come from the normal distribution there, with the mean 0 and variance sigma G squared over M. So where M is the total number of markers, right? So in this particular case, if you add them up, the total variance explained by all the markers, if like assume the markers are independent, will be sigma G, right? So sigma G squared is the variance explained. And the sigma G squared over the variance of Y is something called heritability, right? So this is the model setup. Remarkably, this model has been found to be pretty good, just in terms of explaining the data. And this is the model I'm going to base all the discussion on. Okay, so if you believe in this model, the covariance between two individuals and can be estimated by how similar their genotypes are across all the genotypes. Like C1, this is corresponds to the genome-wide similarity score. The more similar the two individuals are, the more correlated their phenotypes are. So this is very simple, right? Under this model, you can just use a RAMO, standard RAMO, and to estimate, right? And so there are some, like, we don't really know what the causal SNPs are. So there are some kind of use genome-wide SNPs to approximate genotype with similarity. So there's an approximation there. Yeah, so this for one trait, you have this univariate model Y relating the genotypes from all the SNPs. And if you now you can use RAMO to do the estimation. Now you have two traits. You have Y1, Y2. There's a bivariate version of this model can be used to estimate the correlation between the. So I'll come to more later on, but I'll just say the bivariate version of this, the random effects model, can be used to analyze the data to get, but all these require individual data. The challenge for GWAS is like a lot of time you don't have access to individual level data. Even you have, it's going to be very, very expensive to analyze. So the most of the times, much easier to get summary statistics. Again, just remind you what the summary statistics look like. This different rows correspond to different SNPs and there's effect size. Okay, so when you have a summary statistic like this, you don't have individual level data. You do not have access to the phenotype or the genetic or genomic similarities between individuals, right? So in this case, how to really estimate? So this comes to a method called LT score regression, first proposed by Alex Price from Harvard years ago. It's kind of an interesting way just to think about the problem. The LT score regression, I'll go back here and then come back to the figure. It's trying just to very simple to derive the chi-square correspond to the think about the test statistics, right? You look at the effect size. There's a test statistic for each individual SNP to look at the effect size. The null is 0, and the larger this chi-square is, there's more evidence to suggest this one is not it's not 0. And if you truly believe a priori all these markers, they make equal contribution to the trait, and you can calculate the expectation of the chi-square statistics is a function of the sample size. The larger the sample size is, the larger sort of this expected value is. Sigma square corresponds to the variance explained by the genetics, and it's divided by m. So LJ score is called the LD score. What's LJ? LJ corresponds to, when you look at this particular marker, you'll see how correlated this marker is to all the other markers in the genome. Remember that the markers are not independent. So if these markers were all independent of each other, they all have this term just equal to 1. So they should have the same expected, if you believe the model, right? So all the statistics should have the same average, and then things become easy. And if they are not the same, and if there's an interest disequilibrium, and different markers, they will have different dependence on the other markers. And the idea is, if a marker is in a region where there's a lot of dependence with the other markers, so all the effects from the other markers will be piled onto the effect on its own. So the expected value of the chi-square will be larger, right? The degree of the increase has to do with how dependent this marker is on the other. So this LJ is an LJ score to tell you how strongly this marker is associated correlated with all the other markers in the genome. And so the idea then it becomes like, if you do all the chi-square test, and you have on one hand, on the left-hand side, you have all the observed ones. On the right-hand side, this becomes a design matrix, right? Yeah, you just do a very simple thing, and it turns out this seems to be a quite sort of efficient way to estimate the hierarchy, even without using the individual level data. But you do need to have a good reference sample where you can have a good idea how the markers are dependent on each other. So we call the reference genome, the reference panel. Not rather, it's a reference panel to define the dependence among the markers. And so I just said, so far I talked about the single trait analysis, random effects model for trait marker relationship. And we can just use to estimate the overall genetic contribution from markers. Also, you can, one thing to say that now you can look at all the markers in the genome and to see that, but also you can look at chromosome 1 markers, chromosome 2 markers. People have done that to see how each individual chromosome contributes. People found it's pretty much the contribution is proportional to the chromosome size. And also you can put out or look at all the markers in the coding regions, non-coding region. You can do this for all different interesting things, not just only putting all the markers together. And also so this can be used to either individual level data can estimate this using RAMO or LD score regression using summary statistics. Also, this estimate also turned out to be unbiased, even under some model misspecification. What do you mean by multiplication? So this assumption we have made so far is to assume every single marker has effect on the phenotype. But in reality, that's probably hard to convince people, right? You say, maybe there's only like 1% of markers having effect on the phenotype. The other 99% have no effect. It turned out in this scenario, the RAMO is still unbiased, the LD score is still unbiased. And we have a couple of papers, one in Annals, the other in Sinica, to really show that. This is non-trivial proof. But even on those settings, when only a small proportion of markers are associated with the phenotype, the whole framework still gives unbiased. That's pretty remarkable. So that's for one trait. Yes.
So you mentioned genome. So
reference on panel, not reference genome. Sorry. Panel with the people different. Yeah.
And then what we basically need from that panel is sort of a representative of the samples of the animals. Do we need anything else from it?
No. Just want to read the panel to represent the dependence among the markers, right? The LD score regression needs an LD score. I think that's the key. You want the marker to truly represent how the markers relate to each other. If you are off that, then you. And also, we also done a lot of to see the robustness to choosing using different panels, different methods. Some method may be really good, but you have the right reference panel. Some method may not be so good. So there may not, but some may be more right. So there's always like a lot of different methods have been proposed to look at different things. Okay. Yeah. Now we can go beyond one trait, look at two traits. And so for the two traits, we can say you think about this Y as a big vector of the genotypes, Z as another big vector of genotypes. The beta vector corresponds to the effect size across all the markers, and gamma represents effect size across all the markers, right? So for example, beta 1 here may define the first marker effect on phenotype 1. Gamma 1 defines the effect at the first marker on phenotype 2, right? Yeah, now we can really invoke a random effects model, but really quantity effect model for the joint effects of beta and gamma, right? And this term correspond to more for the heritability for the first trait, the heritability for the second trait, and the cross product here correspond to how correlated beta and gamma are. When the two traits they share a lot of genetics, you expect to see there's a good correlation between beta and gamma, which means that if a variant has effect on trait 1, may also tend to have effect on trait 2, and also the magnitude may also be similar, right? So the correlation here between beta and gamma as random effects model really can be used to define the correlation, the genetic as a genetic correlation between the two traits. So this is for the working model we use. It's just an extension of the single trait model to bivariate model, where we still use the same random effects, but the way we quantify or try the way to estimate the correlation there's to just look at the correlation between beta and gamma under this bivariate sort of random effects model. So again, we can also derive this bivariate cross-trait LD score regression. So this is sort of how this is more thing about the test statistics. And instead of looking at the second moment of the individual one, right? So this corresponds to test statistics for the first trait, the second trait, right? If you estimate heritability for the first trait, you can think about this. You can think about this Z square. You can use that as to estimate the chi-square. Is another one. Also, the cross product of the two gives an estimate of the genetic correlation, right? So this also is very intuitive, very straightforward. And also you can use method moments there to gain a little bit efficiency. That you can the detail that is published a while by us. So there's a way you can just estimate using instead of using the score regression, you can gain a little bit efficiency by using the moments estimator. So when we apply this to different traits, it's about 55 traits, you can just see there's a lot of shared genetics across different traits. So there's a 55 different traits here. So there's correspond to the autoimmune disease like eczema, celiac, yeah, the IBD, Crohn's disease. There's a lot of positive correlation. Here, the blue means positive, red means negative. The darker the color is, the stronger either the positive or negative correlation. Star means a significant, right? So you see a bunch of correlated traits. A lot of traits they have positive genetic correlation. Like so, this one corresponds to the psychiatric disorders and this metabolic disorder. You can see a lot of different things. So far so good, right? So we have this way to define the genetic correlation between traits. And now, let's see why we want to go beyond the global correlation. So I'll give you one. First, as I say there, we developed a tool to really look at correlation not at the global level, but more at the local level. In the local level, the challenge is like the markers, there's a fewer markers, and so there's maybe a little bit more careful treatment of LD dependence there. So we have this model called the supernova. That's the localized version of this nova based on method moment. So I'll just skip the details here, just show you the results. When we look at the global correlation there, so in this particular case, each dot corresponds to a particular pair of traits, and the y-axis corresponds to the global correlation, and the y-axis corresponds to if you break down the genome into different segments. In this case, about like 1,500 segments. We can ask whether there's a genetic correlation within each segment, right? And we can estimate what proportion of the segments they show still significant evidence genetic correlation. And yes, we can see here, it really depends on the trait pair. So this may be a lot of due to confounding. It's between the cognitive performance, agitation, attainment. So there's a very strong genetic correlation globally, but also it seems like this global correlation there is really spread out across the whole genome, right? For every segment you look at, there seems to be some correlation there. And on the other hand, for like Crohn and IBD, so these are like, actually Crohn is part of IBD, so it's somewhat meaningless, but you say you can do the GWAS for Crohn, you can do GWAS for IBD. There's a strong correlation here, but in terms of the region, the proportion regions contributed to the correlation, it's much smaller percentage, right? So you can just see there's reason to see there's a like between body mass index HDL you have very strong negative correlation, but also it's contributed from multiple regions, right? For the same genetic correlation across the genome, the number or the proportion segments contributing may differ quite a bit across different trait pairs. Yeah, and also this gives a more like detailed account of like the But now this the example to really show you can sometimes if you look at the global correlation, you may see some counterintuitive results, and that can only be resolved by looking at the local correlation. So this to look at global correlation we see here. If you look at the global correlation between ADHD, attention deficit hyperactive disorder, and cognitive performance, there's a negative correlation there, and it's highly significant, which means that these kids they have the genetic, right, that can put people at a higher risk for ADHD. They cannot pay much attention. Also, they tend to have like negative cognitive performance, right, globally. Yeah, and also you can look at the ADHD and agitation attainment. It's the same negative correlation there. So these kids, I mean, at the phenotypic level, you see that, and also that the genetic level, also you see if a variant put a people at higher risk for ADHD, also the same variant may tend to lower the agitation attainment, right? That's negative correlation means globally. And also you look at the autism disorder and also ADHD, you see positive correlation there, which means if a variant put people high risk for autism disorder also will tend to increase individual child's tendency or likelihood to develop ADHD, right? So there's a positive genetic correlation there. So these are all like make common sense, right? The ADHD. But this part doesn't make much sense, right? So look at ASD and cognitive performance. So you think this is positive, this is negative, expect this to be negative too. But if you look at this, the correlation here between ASD and cognitive performance, that means if you look at a genetic variant on average, if that increase someone's risk for being autism, that tend to increase their cognitive performance, right? It's very hard just to put three different things together. Negative correlation between ADHD, cognitive performance, positive correlation between ASD and cognitive performance, but positive correlation between ASD and ADHD, right? But now if we break down into different regions, we can see that across all the regions in the genome, and for the most part, right, you see the ADHD and cognitive performance. No, it's ADHD and the ASD is a positive correlation. So this is also the same as the global one. The ASD and also cognitive negative is also similar as the global one. So these correspond different patterns. So this ADHD and cognitive also negative ones. But you do see the ADHD and cognitive performance, they have some positive correlations in certain regions. And also for the autism disorder and cognitive performance also have the positive correlation there. Actually, there's quite a few regions locally, you'll see some positive correlation between autism disorder and the cognitive performance, right? So you can see the reason that the global one is really the average across the genome, but locally they do really have different patterns. So when you average them over, you'll see a pattern there that may not really really can miss some important things. But then you can ask a little bit more, let's say, now if I partition, or I try to identify those regions with either positive, negative correlations, are there any signs, any hints that we can get what's going on here? So if we really look at the regions with a positive correlation between ASD and the cognitive performance, and then we can look what these regions are associated for the other traits, right? There's a variance that you can, there's something called the GWAS catalog. You can put in different genes, different regions to see what these regions have been associated with what traits. These are really enriched for association for 10 traits documented in GWAS, including extremely high intelligence, household income, and loneliness. So this region there have these genes, actually, it make people a little bit more intelligent, have a higher income, but more lonely. Yeah, but these tend to be the positive region between ASD and cognitive performance. You may think this may be the high-performing ASD individual that these people. So there are certainly two different subtypes between ASD. There's high-performing one and also the lowly performing one. On the other hand, for the negatively correlated regions, they were enriched for association with a lot of neural development and the psychiatric disorders such as schizophrenia and bipolar disorders. So there are different regions there, and they make a sort of the patterns there that, and deal the pattern between the genetic correlation between these two traits. It's have different distinct patterns, sometimes negative, sometimes positive. That means like you have different groups of sort of variants there. Some these variants people like more like autism, but they tend to be a highly. I mean, for example, on the Asperger syndrome, you can think about is more highly performing one, but it's highly intelligent, but also is more ISD-like. You can really see that. And also if we really partition these individuals, the different regions into the positive and negative regions, positive associated cognitive performance and negative associated cognitive performance, and we can build a score. We can build a score of how likely they're going to develop ASD. So there's a PRS, which I don't have to talk about. You can certainly take all the markers in that region to build a model to predict ASD, right? And so the higher the score, the more likely this person will develop autism. But if you look at their IQ, you can see diverging patterns here. You look at the IQ, look at this. If you look at the ASD, if you go from left to right, these people will have the same trend towards higher likelihood of developing AD. But in terms of IQ, they start to diverge. Okay. So this is the more the genetic correlation here I want to talk about. And the second part, maybe like 20 minutes left, I'm talking about the gene correlation. That's more the genomics end. And so gene co-expression network has been studied extensively in the last maybe 20 years, I think. Yeah, Ali, a lot of people have contributed to this whole thing, the whole enterprise, building the co-expression net based on gene expression data. And the typical flow of the gene expression data is you go to a sample, right? A biological sample, you take out some biospecimen, and you do microarrays in the old days, right? Or now you do the RNA sequencing, and you look at the correlations between these between different genes across all the samples, and you build a sort of a correlation matrix or similarity matrix, and then you build such a network. And then you can do a lot of cool things downstream to look at this network. And this network is so-called modules have been found to really give us some insights about how these genes are related to each other. They also get some functional and mechanistic insight into the tissue and disease. A lot of things, there's something called WGCNA. It's widely commonly used, cited maybe 10 or 20,000 times. It's more like gene module approach where you take the gene expression data, you partition all these different genes into different modules and look at what these gene modules are doing. Yeah, so that's very, very commonly used. But so in the early days, most of these methods are based on the bulk samples where you, as shown here, you just take a piece of tissues out, and that piece may have millions of cells. But the problem is that these millions of cells, they are of different cell types. For example, you look at the brain, you may have the neuronal cells, astrocytes, microglia. This is sort of the cartoonish version of how they look like. Or dendrocytes, pericytes, and endothelial cells. It's very, very rare, but this is a little bit more common. So these are different cell types that can contribute, can really in the sample you look at. So in that case, when you look at the association, you identify a correlation between these two genes in the bulk sample. You don't know what's going on at the cell type level. Yeah, and the question is like, now we're talking about the global versus local or beyond global. Is like the global, I'm talking about the bulk level expression, co-expression, the correlation there. More the local is we'll talk about the cell type specific ones, right? How to really estimate cell type specific ones. And in the last couple of years, we sort of been looking at different ways of inferring this cell type specific network. It turns out that we actually may be able to infer the cell type specific correlation using the bulk samples if we have a good estimate of cell type proportions in each sample. And so I'm not going to talk about today. We have this paper, I think it's on Zhao's website called the CSNet. So if you know the cell type proportions, that can be estimated based on some like a cell type specific markers. You can think the estimate can be very difficult. But if you truly have your hands on the cell type proportions, there's a way you can deconvolve this bulk level co-expression matrix into the cell type specific ones. And so that's one approach. Today, I'm going to really talk about the approach using the single cell data. That's also is the project I think the Y made a significant contribution to in this paper that's published last year in Nature Communication. It's called the CS Core. Okay. So what's the challenge with the single cell data set? It's very sparse and noisy because if at the single cell level, the amount of the genetic material, the biological material is very limited. And also there's a lot of variations across different cells, reflect mostly in the sequencing depth variation. Just give you an example how the single cell data look like, right? This is if you take a snapshot, what's going on. So this is the first like a dozen rows. Each row corresponds to a given gene. You can see the numbers here, mostly 1 or 2. The majority just zeros, right? And this is very sparse. And the other thing is there's a lot of if you look across, this is one of the most abundant genes in data set, long data set, RPI6. You can look across the gene count across all the cells. Yeah, you can see the most time you get 50, but at most you get 50 or little. There's some cell if it's a large, but you don't see many. The majority will be actually between 1 and 10 or 1 and 5. The most cells only this gene. Even this is probably one of the most abundant the genes in the lung samples we analyze. So it's very sparse. And also there's substantial variation in terms of how many sequence reads you'll get across different cells. So this is about like 80,000, 90,000 cells here. And you can see the x-axis corresponds how many total sequence reads you get. Sometimes you get like 500, 2,000, 20,000, but you see that large variation here. So just in terms of the number of reads you get from cell to cell, there's large variation there. And I also can see what I show in this figure is just a projection onto the y-axis, right? But you can see substantial variation just from cell to cell in terms of sequencing depth. Right? Yeah. So how to really estimate the gene correlation from such thing? Very naive one, but you know it's not going to be good. It's just like for every single cell, right? You look at how many times a gene is expressed in this cell and gene 1 and gene 2. For example, for these 2 genes, just 2 random genes. And you can see if you look at the scatter plot, you can just see this gene as observed at most 10 times across all the 80,000 cells. This is not much better, right? And the fact you have just a very limited, every single sort of possibility here, there's a one circle, but this is really there's many cells at this point, right? So but you know this is not going to be good because each individual cell here, each individual sample, they have different sequencing depth. You want to normalize, right? Very naive way to normalize, you divide the number of reads for this gene, divide the total number of reads, right? That's sort of normalized. And then you can see this picture here. And now you see things are to be more spread out. But also you see that each stripe here, actually, they are derived from the same circle, but they represent cells with a different sequencing depth, right? And if you divide by different numbers, you'll have form a line. And if you compare the correlation 0.24, now you normalize by sequencing depth, get to by 0.03, right? So this normalization may not be good. There's many different normalization methods. So there's another normalization method. And still as 0.03, you'll see a little bit different picture, but you can see this. So this is the challenge that we are trying to address, how to really estimate the gene correlation using the single cell data. Another challenge is that what I'm really interested in estimating is the correlation of the true underlying gene expression, right? Every single cell supports an underlying truth. But what we observe is only sort of you sequence that cell and you get a certain sequence rate. And so this is maybe correspond to two highly correlated genes. But because you do the sequencing and there's a lot of sequencing variation, there are sequencing depths, and also you only even if every single cell you observe the same sequence, you still get because there's a lot of sort of the so-called measurement error, right? Going from the underlying so-called truth, you get much lower correlation there, right? So your estimate is underestimate the true underlying genetic so-called the gene correlation. So what to do? Like, so again, like the random effects model, we ask for some help, right? We need to understand what's the correlation here, right? Between this underlying truth, and we want to model in the sampling process from here to here. And the most naive, the simple one, it also turns out to be not too bad approximation to assume that when we have underlying expression level like Zij, the jth gene for the ith cell, sequencing Si corresponds to sequencing depth. And the Sij corresponds to number of times this particular gene is observed, follows the Poisson distribution. Depending on the single cell sequencing platform, the Poisson may or may not be good. For the earlier one, the SmartSeq, it wasn't quite good. People talk about like zero inflated ones. But for the Jobley based, like 10x, it turned out to be pretty good approximation. People have written papers about it. So let's trust this model, right? So conditional on Zij, given the sequencing depth, we can model the Sij as a Poisson random variable with the mean Si times Cij, right? So now we have a model now. And let's see, what can we use this model to really estimate the correlation between two genes? This is only for one gene. And now for two genes or even P genes, we can just assume, okay, let's assume all these different P genes, jointly they have a multivariate distribution denoted by FP. And conditional on each individual one marginally on each one, given the underlying Zij, there's a Poisson distribution there, right? So the underlying, what we really estimate is the correlation between Zij and Zij prime. That's a rho JJ prime. That's our target. That's a parameter of interest, but we only observe Sij, right? And how to do that? And we can just like based on this simple model here, it really pretty depends on the Poisson assumption, right? Based on the Poisson assumption, we can really derive the mean for the expected number. It's simply just a Poisson. We know the Poisson mean, right? And also we know the variance. And the variance here is two components, right? This is the Poisson one, but also there's sort of the actual component here having to do the variation from cell to cell, right? So this one, and also we can write the covariance. Now we have these three sort of equations, the mean and the variance and also covariance. Now we have this observation here. This is observed, and also we have the sequencing data that are unknown. These mu's are unknown. The sigma JJ, sigma JJ prime are known, right? And then you can just based on this model, we can set up this sort of estimating equations that relates the observed the number of counts for each individual gene for each individual cell that defines the mean. We can use this one to estimate the mean. And also we can use the second moment, this one to estimate the sigma JJ. We can do the cross product to estimate sigma JJ prime, right? So this is all good. And also I checked with the White, it seems his idea to use iterative with least squares. It seems, yeah, in this framework that to build each individual equation here, the noise has different levels. And actually this can be estimated using this sort of approximate weights. And that's work out really fast and seem to be giving some very good estimates. Okay. And then build on that, we can also estimate sort of do the testing whether two genes are correlated or not, just doing this the test based on between gene J and gene J prime. Okay. So the idea is hopefully it's clear. We just use very simple like a Poisson model to describe relationship between the observed counts and the underlying gene expression coupled with the sequencing depth, and then use this sort of the moment condition to estimate, to come up with estimate equations and to solve this the least squares problem, but with the weighted least squares. And then we just compare the performance with other methods. Many methods have been proposed to estimate the correlation. So this is an area that has received some attention by people in the field. And so these are some methods. So one simple one just do the Pearson correlation, right? Or you can do some sort of some transformation here, and you can take the residuals and look at the correlation residuals. So this is a case when the genes are independent, and we can simulate independent gene pairs, or we can use observed the single cell data. Think about like permutation lag, but at least a little bit sort of subtlety there. But you can permute the expression levels of gene I and gene J, gene prime to make them independent. And in this case, the independent genes we can estimate. But you can see for many methods here, the estimated correlation the average co-expression is actually dependent on the x-axis with the average, or you think about the geometric mean of the expression level of the two genes. So it's not like independent genes, but here there's some kind of mean dependence pattern there. Whereas the CS core method, also there's a row analytical Pearson correlation seem to be doing a pretty good job, just in terms of identify or inferring, not getting inflated estimate or a biased estimate for the independent gene pairs. But more importantly, if our target is really to estimate the correlation of the two genes at the single cell level, remember, if our goal is to really estimate this one, not this particular one, right? And then we can simulate data where we sort of know the underlying correlation, and we can compare different methods that are not really taking into account this so-called, we can take a measurement error, some noise into account. And in that case, only the CS core can give unbiased estimate. For all the other methods, seem to be a really strong dependence of this estimate correlation as the average gene expression level across between the two genes. Yeah, so that's the importance really, really modeling this so that you can really get to the parameter of your primary interest. Yeah. And also you can think that the time is very fast, very efficient compared to the other methods. And again, this is I think this is the best work. As looking at the real data sets, I think he spent a lot of time going through this, a lot of times worked up to go through different data sets. So there's like 3 brain data sets he looked at, and correct me if I'm wrong. Yeah, so there's this one from Hong Kong, and there's one from MIT, and but to focus on this different data to see for this real data set how to really compare different methods, how to also say, because for the real data we don't know the ground truth, right? So there's different ways that can be used to really evaluate, compare different methods. One is to look at reproducibility, right? If you look at the same gene pair, if it's truly correlated, like for example, for different cell types in the brain, and you tend to recover that in independent data set. So the reproducibility is one way. And so if you look at the ratio, so reproduced pairs between the CS core, and the other one is like this method we chose because it has a well-calibrated sort of type 1 sort of fault-positive arrays here. And this corresponds to just different cell types. And you can just see like different significance levels. And you can do see pretty much this corresponds to no difference between the two methods, and the CS score here really outperform the other methods in that regard, reproducibility. The other one is use some knowledge about the biology. So that the genes are regulated by transcription factors from this known database. You can take the transcription factors and their targets. You really want to identify these pairs between the factors and targets. Yeah, and also you can see between different methods, you can look at the transcription factor and their target pairs to see how many you recover by these 2 different methods, and the CS core seem to do a better job across different cell types. And also you can look at the estimate for correlations. So for the so-called the genes with the similar biological functions. Yeah, and also you can see that they seem to have more uniform, consistent patterns. These are correspond to the goal functions using the chemical synapses and defense response, protein folding, etc. Right. So this, if I have more questions, you can ask the wife. So he did the work. So that's pretty much the second part, is talking about how we can really estimate the cell type specific genetic correlation using the single cell data set. So just to summarize what I talk about, I covered as genetics and the genomics. In terms of genetics, talk about how to estimate the genetic correlation, right? Use globally, either using RAMO, I think about more like using the summary statistics, but also the global this measure sometimes can be misleading, can really mask some variation across different local regions. That's why also we have proposed, although other people have proposed method look at local correlations there. I'll give you an example there for the correlation patterns between the ADHD, autism, and the cognitive performance. At the global level, you'll see contradictory patterns, but this contradiction can be explained by heterogeneity of the local correlations. Yeah, and also I talk about the gene correlation, look at the correlation between gene expression levels. the bulk have been some have been analyzed for maybe 20 years or even more. There's more and more recent interest to look at cell type specific one, especially with the availability of all these sort of the different the single cell technologies and data sets. And also, say why we really focus on correlation. Correlation is such a basic statistical concept. It's often the starting point to talk about the causality, right? If two things are not correlated, there's no point to talk about the causality. So we want to have some good handle on correlation in the first place. Yeah, and also I said the local and the context dependent or this pattern can yield more insights than maybe overlooked by global analysis. And domain knowledge here can be helpful and sometimes are needed to redefine what is local, what's context, the cell type, also the local regions, dependent LD and other patterns. So there's a lot of like details on that. I skip over here, but the most important thing in this age of AI, everything is AI about the machine learning. Data usually noisy, statistical model actually are useful. In this particular case, not any single AI will tell us how to really estimate genetic correlation, right? Unless you have random effects model using framework, you can break it. If without using the Poisson modeling framework, you can never get to the underlying truth. If you believe or what are really doing is really a proper model. Finally, on to thank the people who really do the work on the global correlation. The NOVA was done by Qingshi, developed by Qingshi when he was still in my lab. He's now Wisconsin, the other UW medicine, and the Supernova and Yi Liang, and also even his student. So he's really built on the NOVA and to the local version here. And so also the CS core was really led by Chang, a student, also with a lot of help from the White, and so like published. Also Xin Ning's the current enrolled PhD, and Bill is Cincinnati, and also Emma Zhang from Emory. So this was a group effort of the CS core, and also the cohorts and the funding that support this work. Maybe that will thank you for your attention. Yeah.
Very, very interesting thought. Thank you. Since I have lots of questions,
Oh,
I'll
sure. Yeah.
contain my excitement. So you mentioned that with bulk data, if you know the proportions, you'd be able to sort of decompose it.
Yeah.
In what you presented, you were directly estimating using single cell data.
Yeah, you can do both ways. Yeah.
And so that's what I was going to ask. If sort of if you are estimating using single cell data, I'm assuming that sort of for each cell type you're estimating separately.
Yeah.
But then there's no connection between those networks that are being estimated for different cell types.
No.
Whereas if you estimate the qual, those networks would be connected in the sense that they're summing up to become that sort of overall network. Are there any reasons to think that sort of we would want to do joint estimation of the network across cell types?
Myself, I may be biased, but I tend just to probably put my more efforts into single cell base, because it's very difficult to estimate cell type proportions. I mean, like it's impossible for like rare cell like endothelial, it's almost impossible. These are very, very difficult to estimate, and unless you have very abundant cell types, it's very difficult. Now without the single cell data, I'll just go straight into single cell based.
Maybe a quick follow up on this. So I totally agree.
Yeah.
Sort of use single cell, which increasingly single cell is available. But even if you're estimating with single cell, you can estimate there are two different ways that you can estimate. One is that sort of each cell type you can estimate separately.
Sure, yeah.
Without thinking that sort of the gene for expression networks across cell types are somewhat similar. The other is to again do another level of sharing of information across cell types.
Right.
Say that sort of potentially the gene expression for expression networks across cell types are somewhat similar.
Yeah, in another dimension, actually, the White spent some time, but that project got stopped when he was with us trying to look at individual cells.
Yeah.
So instead of looking at different cell types, he was thinking about different ways. Now he's talking about like single cell data set. But if that is because data set comes from 10 or 20 different individuals, yeah, he can resume with you, but sort of, but there is, he actually put a lot of effort to think about that, how to really try to analyze data from multiple individuals and to estimate the cell type specific. But of course, if you have different cell types, it's another level you can integrate, right? And also, so you can make it very, very complex. When you have the spatial single cell data, the spatial data, you can also look at the spatial. So there's it's a question like what we're trying to capture here, like why we are interested, what kind of thing we're interested in looking at the co-expression patterns. Yeah, the co-expression, any correlation there is a population average. You can break that down into finer scale at what level you want to break that down. So it's always aggregate something. Yeah.
I guess, I don't put it into account the like genomics of that, but I guess I thought it more about the type of modeling and methods to give it. Like, I guess my understanding for the first part we use randomized method like.
No, there's when we have individual level data, you can use REMO. Not we can use the meta moments or like yeah.
So basically like but the second part also.
Sure, yeah.
I guess it appears to me it has some connections with things like the phase, the phase becomes still not there.
Mhm.
But I guess there's some. But we probably know also is like the original like the phase. I would pick these papers is actually about like fossil times.
Right.
I feel that's what it's about.
Okay, yeah. Thanks for the we can talk more about it. Yeah. Yeah, sure, yeah.
So maybe going, you mentioned something about this at the end, in the first part of the talk. So what we presented seemed to be like related to Simpson's paradox, in the sense that when we aggregate,
Yeah,
we
that's
draw a different.
absolutely right. Sure, yeah. Yeah, the whole thing. Yeah, that's the whole right. It's a global versus a local or something. It's a Simpson. Yeah.
In one way, sort of we could get around Simpson's paradox. It's like thinking about sort of causal relationships instead of just correlations. And so I think you alluded to at the end, so that sort of correlation is the first step.
Right.
Given that sort of genetic effects, we could think of them as sort of naturally randomized. So we see directions to sort of think about sort of causal effects of genes, and then sort of thinking about sort of instead of thinking about correlation between outcomes, thinking about sort of causal model.
Yeah. But the circle, the whole year, the Mendelian randomization, right? Yeah.
So do you think that could be sort of another way to think about sort of this sort of in a sense deconfounding what the effect of across the whole genome is? Is that sort of a different direction to think about this?
It's interesting, yeah, because it really depends on for the Mendelian randomization, depending on the instrument variables you're using, right? And I think in that regard, the correlation or association between for that causal, we are asking different question, right? Between exposure and outcome, and whatever, regardless, as long as the instrument variable either it has a positive, negative effects, it's going to be reflected through, even purely through the exposure. It doesn't depend on which instrument variable you use, right? So you'll have heterogeneity. Some variants may be increasing the risk, maybe decreasing the risk. But I see your point. If there's negative correlation there, that's something you have a direct path. So that violates the assumption about Mendelian randomization. And there's a people like trying to like make this method more robust in the presence of sort of direct path, right? The horizontal effects by using this global correlation there. And if the global correlation is misspecified, then there may be a problem. So if there's no violation of this direct path from the instrument variable to the outcome, then there wouldn't be a problem. But if there's indirect path, some of these methods that have been proposed to capitalize on the correlation between, or the genetic correlation between this exposure and outcome will have trouble. Yeah. Then that's going to be problematic. Yeah.
I want to ask this question as well. I think the first part of the talk is like local global. I think it's very cool. It's related to Mendelian randomization ideas. So like basically like all the heterogeneity could be due to a lot of reasons. One type of reason is that this local gene is for this trait. So it's estimating something related to this trait's effect on that trait. And some local genes for that trait. So it could be related, estimating the causal effect of the other trait on this trait. So basically their correlation could be, like the opposite sign. That's totally possible because of that. And also you can have some local genetic regions that are like influence both traits, and then you're estimating like confounded relationship or biased relationship due to a pleiotropy. So I think you were trying to decomposing that without explicitly saying.
No, I think this sort of the presence of heterogeneity there is going to be a problem for Mendelian randomization, because it depends on the trait pair looking at the exposure and outcome, right? So if there's a genetic correlation between exposure and outcome, unless it's purely through the mediator effect, mediation of the exposure, if there's a direct path, and any effort to use that direct path will be dependent on which region, which instrument you use. So that's going to be issue that can give you some headache or some maybe bias estimate or the wrong estimate of these things. Yeah. So it's to be very careful about it. Yeah. So the choice. So this is going to be problematic. Yeah.
Totally agree, and it relies on having a genetic region where I'm more or less sure that this is only influencing one trait, not influencing both traits or so I think it totally rely on that.
So you may end up maybe in a scenario, if you use different instrument variables from different regions, you may get different results, right? That mean even when you try to take into account sort of the horizontal effects, yeah, on outcome.
Yes, yeah, and for traits like I know I have 6 SNPs, which I am more convinced that's specific for one trait. Then I'm more sure like this is estimating the I have more confidence in that local region. I guess that's what I'm saying.
Right.
And therefore the other regions I'm less confident they're estimating something related to causality, but for this region, because it's specific for one of the traits, then I have more confidence that it's estimating something related to causality. And
yeah, I think I mean, the concern is try for example, for this example, the autism, ADHD, and the cognitive performance, if you use markers from different regions, for those methods that try to take into account the sort of global or some local, you may get contradictory results. Yeah.
yeah, we can talk more.
Sure. Yeah. Okay. Yeah.
Any other questions? Thank you so much.
Well, thank you. Yeah. Thank you.
Yeah, I think that's like my point is they. So for Mendelian randomization, we cannot